So we're ready to look at the last part of this chapter. And I have to tell you that your textbook does not go into the detail about the pancreas and its important role in terms of balancing blood glucose levels. So I'm going to add stuff to this part that your chapter doesn't cover. I have put pretty detailed notes in your note outline to help you along here because it's not written in your textbook. So let's get started and let me tell you why it's so important. The big reason why I emphasize the pancreas so much and its role in balancing blood glucose is because of the drastic increase in type 2 diabetes in this country. I think it was about 10 years ago, I was living in Hays County instead of here in Austin, when I saw a study that was done by the Tri-County area hospitals and health centers. They took a look at the rate at which obesity and diabetes had increased in the three counties, Williamson, Travis, and Hays. And then from there, they extrapolated and predicted what kind of change would be likely over the following 10 years. Now, again, I lived in Hays County, so that is the number I remember. And I was shocked that they were expecting within 10 years for the number of people to develop type 2 diabetes to increase by 852%. That's a huge increase. And that is about the time that I started to say, all right, if I'm going to be teaching this stuff, let's make it pertinent. So let's take a look at the pancreas's role in blood glucose balance, and also let's have some frank discussions about diabetes. Here's a map that was put out by the Center of Disease Control a few years ago. But it shows here at the top the trends in obesity in this country, and then below here, the trends in diabetes, in particular type 2 diabetes. And you can see here that there has been an increase in obesity in this country. Diabetes seems to be stepping right behind it in terms of its increase in occurrence. So what I've done here is I've covered the years that were posted by CDC, and I want you to think about this. Let's guess the years. Is this first set of maps, what is it, 1930? Maybe 1950? Maybe 1970, who knows? What would this one be? And this, how current is that? Well, on the next slide, we take a look at those numbers. And here they are. So our last numbers that are looked at here were just 3 years ago, and the yellow font is not showing up well. So I'll write in, this is 2015, this is 2000, and this is 1994. So essentially, over the period of 20 years, our country has gone from being a little obese with some diabetes, all the way to most of the country experiencing quite a bit, more than 6% of the population is obese. And here, more than 10% within these southern and western states have type 2 diabetes. That is a dramatic increase. And again, when I saw these numbers, this is when I said, let me include this stuff in our discussion about the pancreas, insulin, and glucagon. let's look at this slide in terms of anatomy first. And so the pancreas, which is tucked up underneath the stomach and has its secretions, exocrine secretions into the gut, but the endocrine secretions from the islet cells are hormones, and therefore they are carried by the bloodstream. The liver is included in this picture because it is the primary target for both insulin and glucagon. This is a photomicrograph of the pancreas, and the majority of what is in this picture are referred to as pancreatic islets, and we would be able to outline that islet collection of cells here, and again, those are endocrine. You may ask yourself, what are all of these other cells that are out here? Well, these are referred to as acinar cells, and they are exocrine. They secrete into the gut to help with digestion. We will look at these pancreatic cells when we study the digestive tract. So let's go back then to our pancreatic islet cells, and we study the two major ones, alpha and beta cells. The alpha cells produce glucagon. The beta cells produce insulin. We are going to look at these two hormones and their physiological effects in more detail next. Here is another image from the old textbook that I used to use that shows you that in essence, insulin and glucagon levels in the bloodstream are on a teeter-totter. The other thing that is pointed out here is which hormone is dominant in the fed state versus the fasted state. And so in your notes under item E, blood glucose balance physiology, I'm going to give you a little background that you would have learned if we studied metabolism at this point in time. We don't. I talk about it later when we study digestion, so I need to give you a little more information. So the absorptive or fed state, this is as you are eating and immediately after. Insulin is dominant because what you are eating are carbohydrates, some simple, some more complex, and if you like white sugar, table sugar, you're eating glucose and sucrose. So at that point in time, your blood glucose levels will increase because you've eaten. Insulin is dominant at this point in time. We're going to take a look at the effects of insulin in just a minute. Let me give you some background first on the fasted or post-absorptive state. This is the condition in which your body finds itself somewhere between 4 and 6 hours after eating. At this point in time, your blood glucose level has dropped. When that occurs, glucagon is your dominant hormone, and it will trigger a release of more sugar into your bloodstream from stored sources. Let's look at that now. Using the same slide, what I want you to now focus on, instead of our definitions of fed and fasted state, is the effects that our different hormones will have. Again, when blood glucose levels increase, that results in a release of insulin. And insulin has these 4 key effects. And when you look at it, we can see a trend of storage of nutrients. Glycogen synthesis, fat synthesis, and protein synthesis all means we are building bigger molecules from monomers. Glucose oxidation, if you'll recall, this is something that happens in the cell in an attempt to make ATP. So high blood glucose levels are going to influence this feedback cycle, result in an increase in insulin. Another thing that can trigger it besides high blood glucose levels would be high amino acid levels. So if you ate a lot of protein that was broken down into individual amino acids, that also will trigger the release of insulin. Now, another thing that triggers the release of insulin is, well, we call it a feed-forward response. If you put food in your mouth and swallow it and it goes into your stomach, that triggers your pancreas beta cells to release insulin, as though it's saying to your body, get ready, here comes food, let's release some insulin so that we don't have a huge spike in sugar, but that as soon as the sugar is absorbed, insulin is going to help store it. Also, when we are just in parasympathetic mode, we look at food, we think about food, that also triggers a feed-forward release of insulin. Get ready, here comes the food, we're going to need to store it. So when we take a look at the effects of insulin, again, you can see 4 of them over here. I have also listed several things in your notes. Now, besides increasing synthesis and storage, insulin also will inhibit catabolism. In other words, it inhibits the breakdown of these molecules. So it's going to inhibit glycogenolysis, gluconeogenesis, lipolysis, and protein breakdown. All of those things are breaking polymers down into monomers, and insulin inhibits that. So one part of insulin is to stimulate storage, and the other part is to inhibit breakdown. The overall effect of insulin is ultimately to lower blood glucose levels back to normal within the set point. Here is a table from the other textbook that outlines a number of characteristics of insulin, where it comes from, its effect, its half-life, etc. So take a look at this and see if it makes sense to you, because we're going to do some activities that are going to tap into your knowledge here. So hang on to this table. Again, it's not in your textbook, so you may want to either take a screenshot of this slide or a picture with your phone. I also am going to have copies for you in the classroom. One more slide regarding insulin, and again, it is boosted within the fed state. So here you see that you eat a meal, and these are all of the mechanisms, the pathway by which the beta cells of the pancreas will produce insulin. And then this part is the effects of insulin on various target cells, and then overall, what is the ultimate result. See if you can follow this, and if not, ask me questions. So let's go back to this slide, this time looking at the bottom of it relative to glucagon. Again, glucagon is the dominant hormone in the fasted state, 4 to 6 hours after you've eaten. Now, what prompts the secretion of glucagon from the alpha cells in the islets of the pancreas? Low blood glucose levels. Also, if there is a low level of fatty acids, that will prompt it as well. In other words, your blood levels of your nutrient molecules that your cells are going to rely on are low. But here's a weird thing. You remember I told you that high amino acid levels will trigger the secretion of insulin? Well, high amino acid levels also stimulate the secretion of glucagon. And this is why. If you eat a very high protein meal, there is not sugar in it, or if there is, there's very little. So again, the feed-forward mechanism as you start to consume this meal will cause your body to secrete insulin. The overall result of that could be a dramatic decrease in blood glucose levels. Remember, you're not eating sugar, you're eating protein, amino acids. And so the pancreas co-secretes glucagon in that situation. Insulin lowers blood glucose, glucagon raises blood glucose. And so when you eat a high protein meal, the effects are modulated by each other. And this is what will keep your blood glucose from dropping dramatically when all you're eating is protein. Now, what are the effects of glucagon? I have 3 key ones over here on the left of this page. I've also listed several others in your notes. Overall, glucagon shifts your body to catabolism. Glucagon is going to cause you to break down stored nutrients. Remember, insulin caused you to store it. Glucagon will cause the breakdown, and thereby putting glucose into the bloodstream. And the overall result then of glucagon is to increase blood glucose levels. So I'm hoping that you can understand why the author of this textbook, from whom I use this slide, shows it as a teeter-totter, because your insulin levels and your glucagon levels teeter back and forth based on primarily your blood glucose levels. Here is a similar table that I showed you for insulin. This time we're looking at the characteristics of glucagon. Again, either screenshot or photo of this would be helpful for you. I'm also going to hand out a copy of this in class when we go to use it to problem solve. So let's look at this concept from a slightly different point of view. Let's look at variations, changes in your blood or plasma glucose level relating to changes in insulin and glucagon. So we're looking at changes over time, as we often do in physiology. And here on the top, we have the plasma glucagon concentration. The lower graph is plasma insulin concentration. And then on the side, we have essentially another way of looking at these numbers, the plasma glucose concentration. Now, what is normal blood glucose? Some doctors will say 100 to 125 milligrams per deciliter is normal. I would say that when my blood glucose level gets at fasting, this is at fasting, gets down to 80 or 90 milligrams per deciliter, they're really happy with me. And so I think these numbers over here, the medical community is starting to lower those values. Say 90 to 120 or 90 to 115 milligrams per deciliter would be considered normal. So let's take a look. When we have a release of plasma glucagon, when does it happen? So this is glucagon. As soon as you eat a meal, look at what happens. It drops. But over time, after about 90 to 120 minutes, it's going to start rising. Why? The dotted line is your blood glucose level. It starts lowering. So this slide, or this graph, shows you the relationship between glucagon secretion and blood glucose concentration. The lower graph shows you what happens concerning insulin. So look, here's your meal, all the way down here. Look, it starts increasing before you eat, before minute 0. We start to see a spike in insulin, and then after the meal, it begins to gradually drop. Several slides back, we took a look at the effect of insulin during the fed state, right as you are eating and immediately after. This shows you the pathway, the mechanism for hypoglycemia. In other words, you are not only in the fasted state, you are fasting. You're beyond the fasted state. So again, we have a decrease in plasma glucose levels, and you can see the changes that occur in terms of what gets secreted and broken down and why, and then ultimately, what is the result, an increase in plasma glucose. I include this slide just as a warning, preparation for the fact that we will be doing a blood glucose level lab during the laboratory time. And this is just a picture of one way to go about doing it, to test the blood using a glucometer test strip. Our system is a lot safer because we don't have the test strips just loose. It's within a glucometer. So this is a pretty cool lab, and it really demonstrates a variety of issues that we've talked about in terms of blood glucose balance. All right, let's go back, and it's back in your notes under insulin. Let's talk about disorders, in particular insulin disorders. And diabetes mellitus is the domineering disorder in terms of insulin problems. So the normal subject would show a change in their glucose tolerance test, where their fasting may be 90 milligrams per deciliter. They consume a concentrated glucose syrup, and it goes up to about 125 milligrams per deciliter, and then it normally drops back to 90 or below. But the diabetic individual will start with a fasting blood glucose level above 125. That is considered to be an indication of diabetes. And after they drink the high glucose concentration syrup, the oral glucose, then it continues to climb and continues to climb. And that indicates diabetes. So here below are really nice clinical criteria for diabetes. We are going to talk about this in more detail in class, but to understand the difference between diabetes mellitus type 1, which is also called diabetes mellitus insulin dependent. This is often referred to as juvenile onset. And what happens with this is the beta cells of the pancreas are, for some reason, damaged. Now, it could be genetic problem, or it could be the result of a childhood disease that resulted in high temperature. Sometimes a high temperature, high body temperature, we know can denature proteins, and those beta cells have proteins in them, and so those cells can be damaged by high fever in early childhood disease. The key to diabetes type 1 is that these patients cannot produce their own insulin, and therefore they need injected insulin. They depend on the ability to inject the insulin. How does this contrast then to diabetes mellitus type 2, which often is referred to as diabetes mellitus non-insulin dependent? These patients, we used to say it was adult onset. So quite frankly, we don't use the term juvenile or adult for either one of these conditions because, unfortunately, we are seeing more and more kids with type 2, probably due to lifestyle, diet, the consumption of high glucose corn syrup, the lack of exercise, the increase in obesity in kids, all leads to the development of type 2 diabetes. Not in everybody, not in every kid, but there is a correlation between diet, exercise, and consumption of a lot of sugar, a lot of processed foods, and the development of type 2 diabetes, regardless of age. So the key thing here is the type 2 diabetics. They make enough insulin. Their insulin is good, at least at first. After several years of the condition, it can decrease. So they have plenty of insulin. What is the problem? The problem is the target cells, the receptors on the target cells are no longer sensitive to the insulin, and they don't respond. And therefore, it won't trigger glucose uptake into the cells. This slide and the following slide, which is similar to it but from the other textbook, is how a decrease in insulin production can trigger pathology. And I'm not going to go through this entire slide here. I'm not even going to go through it in lecture. I'm going to point out a few things about it in the next slide in class. I will promise you, though, that at the end of the semester, after we've talked about what the kidney does and how the pH is affected by various things, we are going to take a look at this slide and the next one again towards the end of Unit 3. Okay, as promised, I've put this slide again at the end of the chapter. And just as a quick review, we looked in Part 1 and a little bit in Part 2 as well. We looked at hormones, the general pattern, characteristics, what do cells do in response, etc. We started to take a look at the hypothalamus, its relationship to the posterior and anterior pituitary, and some of the hormones that those glands produce. Part 2, we took a closer look then at these glands and some others. And then Part 3, that was the pancreas and its effect on blood glucose levels. So go through this and say, okay, could I look at my objectives, and based on learning these things, can I answer the questions in the objectives? Your first time through, you're probably going to need your notes. But to be prepared for an exam, you should be able to answer all of those objective questions without looking at your notes. Now, if you need help understanding how to do that, who are you going to call? You're going to call me. Make an appointment with me. Remember, I love helping students.